with Stephen Allison, who's a LEED certified and ISA certified arborist, landscape designer with Flora Teeter Landscape Architects in Baltimore, Maryland. So, welcome, Stephen.
Thank you. All right, again, my name is Steve Allison. I graduated from West Virginia University, and I now work for Flora Teeter in Baltimore. Okay, so this is challenges of maintaining living systems. This is a niche field of my first lecture at Engineering Green 2014 in Baltimore that covered urban bioretention. Are we there yet? I am missing a crucial piece. Clicker. Okay, so we're mainly going to be talking about urban bioretention. Soon. Okay, we have a problem with our bioretention facilities. We are getting all the funding, we have all the science and all the information that make these wonderful facilities. We're capturing Mother Nature and bringing it to our streets and our highways to clean the water that we have dirty, that we have polluted. But we haven't taken the time to figure out how to maintain these, how to create a system that regenerates, that holds itself. So there are three topics, there are three pieces that we can look at on how we can make this bioretention work in a longer amount of time, stay revitalized, stay energized, and complete in the soil. So I'll be covering these three goals that we need to understand our system, to maintain it, to preserve it. We'll get there. Okay, it's establishment, development, and preservation. When we come up with the idea that we're going to use a bioretention facility or constructed wetland, we have to look at why we're creating it, how we can develop it, and how we can preserve it. Okay, bioretention facilities, as I've said, it's Mother Nature's way of filtering it, and scientists and research in the 1990s had figured out that this is an ingenious way to clean our water in a relatively low-cost manner. In Baltimore City alone, in their watershed implementation plan, for bioretention and constructed facilities that are going to be done in 2018, there's $15 million allotted for this, where they want to take 265 acres, convert it into this area, constructed wetlands, bioretention facilities, and they want to pull out 300 pounds of phosphorus, 1,800 pounds of nitrogen, and 108 tons of total suspended solids a year out of these facilities. $15 million, 265 acres, 108 tons of total suspended solids in our systems. We're not there yet with maintaining them, and we're starting to feed 265 more acres of it in the city. This is part of our problem. Okay, this is on my way to work. This is a facility, the great pilot project, and this is the before picture. We've done everything right here. Native species, the soil ratio, correct location, the velocity is correct. We have trees filtering into the system. This is when it was built. This is today. We have lost the organic material. We have a plant monoculture. We've lost the metabolic soil horizon, which I'll talk to you about. That's a term that I'm kind of starting to use to describe the living environment in the soil that's actually doing the function, doing the work of the bioretention facility. Eroded media composition, like I said, it kind of turned it into all sand. All the light organic material actually went as sediment right into the bay. Again, harboring invasive species, organic matter loss, create an incubator for the invasive species in all these other abandoned lots in Baltimore City. Now you got your place that's going to hold that seed bed until we correct it. All right, here's another example of where we were a little bit off with the maintenance and organic compounds going in these facilities. We're having to irrigate them. We're irrigating native plants in our bioretention facilities. So not only are we creating a facility that's not functioning, now we're putting potable water into the system just to keep it alive. we can accomplish more. Establishing the metabolic soil horizon within the bioretention facility. Bioretention facilities have a ratio soil mix, a medium mix that we use that's based on 24-hour inundation, and it has an organic layer, but it's usually somewhat small. We want to create a horizon that has a chemical exchange in it, and that's where we first want to start with that. We can create a habitat that is biodiverse. This will keep your soil there, dense planting, and affect the denitrification, absorption, and also have aesthetics. You will have this structured prairie view, not a overgrown grassland. It's possible with site design. And then another example of a successful facility, and a lot of this comes down with maintenance in the first initial care. When you develop these facilities, they're almost like a baby. You have to go in and do 6-month checkups and maybe 8 checkups in the first month. It's kind of like that. This is a biological living system, not an aesthetic planting. It just happens to look nice. All right, a couple takeaways. The metabolic soil horizon, plant density, right facility, right side. Now, biological development. So what we've just covered is why we do this, why it's important, what we're kind of losing. Now we're going to talk about the biological development. What is actually going on? What are bioretention facilities? A lot of designers kind of look at plants are solar-driven pumps. And we put them in, and the magic goes that the water goes in, pollution comes out, we're done. And the water's slowed down. Well, there's more to it. The actual mechanism within bioretention facility is that organic layer at the top of the bioretention facility. Right below the mulch, right on that thin line, metabolic horizon where the roots are working. That's the hidden guide, that's the magic. Okay? And a lot of these facilities were worried about the plants surviving. But are they taking the nutrients out of the water? Do they have that layer? It's about the chemical exchange. That's what's going on in there. The whole reason bioretention works, because we figured out that through cation exchange and other chemical compounds decomposing in that organic layer, we were able to, not to get too much chemistry, break apart the nitrogen and the phosphorus. The plants did this with their roots. But there was other things going on. Mycorrhizae, talking about the fungus layer. This is very important. In my field of landscape architecture, we don't talk about this. We look at the list of plants that do well in drought tolerance, and we'll take that flood. But the whole reason it's there is to take the pollution out of the water, to cool the water, to slow the water into our system. That's why we don't have a big chemical plant. This is the chemical plant. So we need to think about that. Okay, here's mycorrhizae. Here's the fungi. Here's the macrobiota that we're looking at, or the microbiota, I'm sorry. Arbuscular, mycorrhizal fungi. You can find this everywhere. I have a couple pictures from my own test gardens in my yard. We'll go on further. This is not my photo. I like my photos a little bit better. But these are the roots of roots. If you look right here, that's your root. That's your native plant growing. If you don't create your metabolic soil horizon, that's all you got. That's all that's taking out of the, taking the nitrogen, taking the phosphorus, taking the pollutants, the suspended solids. That's all that's able to hold that soil. But if you create that organic layer, and you hold that organic layer, you create all of this, almost reaching the plant's path by like 150% more you're covering. Now, if you have 80% sand, if you have 5% organic content, you don't have any of that. You don't have the thorough process of decomposing the organic compounds. This is something that we're just starting to learn about. I've gone and started researching soil scientists' papers from Cambridge, from MIT, finding this information. It's out there. Just hasn't connected to our field yet, not in an applicable fashion. All right, this is my route from my yard. I cut this out. I told my wife, I was like, I can't just show pictures from someone else. I need to show it from my yard, the mycorrhizae that I've grown in the last year. And I had no idea if it was there. This was kind of a risk while the kids were sleeping. So I've kind of pushed back the mycorrhizae and had the root ahead of it. I shook out and washed out the soil to try to get a better picture of, am I right? You know, I see this, but is the mycorrhizae really making a difference? Is it holding the soil? Is it really creating that much more? Or is it just a fungus that grows in this symbiotic relationship and it just lives? Okay, you remove it. Now the beautiful thing of this root itself was only about like that big, two-pronged. When I finally got it out, you couldn't see in the 2D picture, that mycorrhizae system was like a 3D ball that it was able to capture that much more pollution and nitrogen and start dissolving those essential minerals so the plants could take it up. Very interesting. This is the game changer. This, I think, we will start looking at inoculation of these bioretention facilities to grab this and to hold this. This will create that type of microbiota that will essentially create more pores in your soils. You're going to get more earthworms, you're going to get more microbes and microorganisms living in that area because of the release of different enzymes in this fungus. So now you're getting that topsoil, which we were not necessarily told we could put into a bioretention facility. Now it's becoming porous because our system's working. So not only are we creating more of a biological process, we're actually making it filter better while letting it last. And there's another picture. This is from butterfly milkweed. This is the first picture I pulled up. The beauty of this type of fungus and paying attention to it in our bioretention facilities, it's one of the few microorganisms that we can actually see. You can actually take your shovel, and this could be part of a maintenance crew, take it and look. Do you see white? You can mark that. This isn't something we need to take and test. You can see if it has that AMF going on there. That's literally 4 inches. And you see that it's growing in more of the dark organic compound than until you get to that other soil horizon. So the thicker organic layer is definitely the way to go. All right, another point to bring in when you're developing these types of fungus symbiotic relationship within the soils that is so essential. We have to remember what bioretention started out at, what it initially was studied by the soil scientists and extension services at universities. And it was terrestrial forest communities. That was optimal. Now, we can't do that on all our curbside bump outs, and that's understandable, but remembering that it just doesn't have to be grass. It doesn't have to. It can have shrubs, and there's a benefit because you'll get different type of microbes with different type of species. And it automatically creates the chemical process. Probably early, like 5:30 this morning, I was going to tell you inoculation wasn't needed. But I was still doing work on my project before I got in the car, because if I tell you something wrong, it's not going to help anybody. I think inoculation could work, and I'm going to get into it further. I read a new study before I walked out the door, so you're not going to see a slide on it, I'm going to talk about it. But it does create it naturally. Every single test bed that I've created with different plugs from North Creek Nurseries, they were very nice to let me grab like 50 of them from them. They had all this fungus already, immediately. I didn't do a thing. All I did was put in some compost, the normal way you do a bed. And all the water was going through. There was no sitting with all that rain we had last year. And again, another example of a living system that we can do that type of biodiversity. That soil and the microbes in there are probably pulling so much more pollution and holding on, sequestering pollution, than something that was simply turf at a bioretention or just the limited monoculture. Takeaways from that, the cation exchange, having the clay, that is essential. These things are studied for a reason because they're literally pulling the nutrients out of the soil. They're preventing leaching into the sand. Without, not to get too chemically involved here, but without having cation exchange, and you're not holding, you don't have a certain kind of organic layer in your soil, you're just mostly sand, the leaching of chemicals will happen because of the chemical process. They don't have enough to hold onto in sand. Sand has a negative charge. So no matter what you put in there, the sand will leach it more to the watershed than from the organic layer. 2-hour infiltration rate, and again, the metabolic soil horizon, very important. Okay, so I've hit on what's really going on in the bioretention facilities. Okay, it's the organic layer. That's your machine. How are we going to preserve it? That's the big deal. That's what I wrote on my abstract. How can we do this when budgets are cut? Maintenance of mulch. It doesn't always happen, like we see in one of my earlier slides. Well, first of all, if we're going to figure out how we maintain it, where did it come from? Where are we getting the biology? We talked about it, but where's the biology naturally seen? It's actually forested wetland biology. The EPA, it says itself, bioretention areas should be vegetated to resemble terrestrial forest community ecosystem. We're not going to build a forest in Baltimore, in New York, but we're going to try to create that biology. So, okay, that's what we're trying to create. What would go on in something that's been growing for hundreds and hundreds of years? And we bring it there. Okay, this is our preservation issue. This area has huge microclimate swings. If you have a bioretention facility on this side, it may need a totally different type of maintenance than on that side, depending on your solar gains, depending on how much velocity of water. When we put these facilities in, construction projects happen then up the street. We never change our facility. Then we get that first flush erosion, and we don't go out and necessarily check it. We don't follow the guidelines. We lose that system. One of the problems with these systems too is bioretention facilities are never neutral. They're either helping the environment and assisting the environment, or they're hurting it with compaction, standing water, area for mosquitoes, or the water is rushing around that facility and then having higher velocity in our stormwater sewer system. So, we need to have the preservation on the top part, on the beginning, on the infancy of it, before it gets too late, because all those days that you're waiting, months and years to go and fix it, we've lost that for the bay, and then you can get another F rating, like we did in 2013 for the Patapsco area, for the Chesapeake Bay. So that starts up the preservation plan. This is an example of what I think we can go off of as designers to try to submit to agencies, municipalities, get published. This is a portion of Seattle, Washington's Green Streets Operations and Maintenance Facility. They are doing a wonderful job with this type of maintenance program that I would like to bring to the East Coast in a hybrid version. It's, again, the Intelligent Service Plan. And what it does is it looks at different facilities. Instead of having a huge budget for landscape maintenance, it looks at separate service areas and how we can take our facilities and put them into blocks instead of having a big landscape crew just come and do every single street. We'll take pictures and show what service rating we need. All right, so if it's missing this amount of mulch or it's missing this amount of plant life, it will get service level A, and we have a price for it, and service level B. If it's a system that isn't getting affected by certain solar gains or microclimates, then we can grab service level B, service level C. And this can change throughout the life of the bioretention facility. It takes more time in the beginning, but you gain that much more biological activity by addressing what needs to be addressed than flushing it, having to replant it, losing everything 1 year, starting all over again, losing your biological process. So I think this is a very interesting way to go about it. I think it needs to be hybridized to our setting in the Northeast, but I think there's a lot of potential there on a way to individualize our maintenance plan in that type of manner. And we can integrate these into our natural setting and have them clean, have them full, deep, and rich